Thank you very much. It's an honor to be here. In this project, our group actually focused on a second barrier within the brain, not the blood-brain barrier, but something called the brain interstitial barrier. Essentially, this barrier is formed by the brain extracellular space, which you can see outlined in red on this electron microscopy image. The extracellular space makes up about 20% of the total brain, and it's been modeled as a series of fluid-filled pores with an average size of about 100 nanometers. Different components of the extracellular matrix form these narrow corridors that limit the movement of compounds within the brain. This has the potential to have broad implications for different biological processes, for drug delivery within the brain, and potentially for clearance through the gliolymphatic system. We've previously seen that by delivering therapeutic ultrasound in pulses, you can widen the extracellular spaces in tissues ranging from flank muscle to tumor xenografts. These effects are reversible, but allow locally delivered and systemically delivered nanoparticles to achieve larger volumes of distribution. The theory is that the radiation forces that are produced during sonication cause maximal tissue displacement at the focal point, with significantly less displacement immediately adjacent to it. The steep gradient produces strain within the tissue, resulting in mechanical disruption of the structures that occur at the cellular interfaces. Our hypothesis was that by a similar mechanism, pulsed ultrasound could be used to generate non-destructive structural changes within brain tissue, producing large pore sizes within the brain extracellular space. We obtained brain slices from young rats, and we used a customized chamber to couple the slices to the ultrasound transducer. We then, after sham treatment or sonication, performed electron microscopy to study the ultrastructural changes that were taking place in the brain parenchyma. We saw that in the ultrasound-treated slices on the right-hand side, the extracellular spaces, which you can see at the bottom, and particularly the perivascular spaces at the top, were larger and more prevalent than in the corresponding sham-treated slices on the left-hand side. Using a MATLAB script to quantify the spaces in each image, we confirmed that the area taken up by the extracellular and perivascular spaces were in fact larger in the sonicated slices. Unfortunately, electron microscopy is not always the best technique for measuring these spaces in the brain because of artifacts that can be introduced by both tissue ischemia and tissue processing. Another method involves studying the diffusion of different sized molecules within the brain, and by comparing the actual diffusion rates to what you would expect, you can estimate the pore size of the tissue. So here, after sham or ultrasound treatment, we injected fluorescent nanoparticles into the cortex of the brain slices and used high-resolution confocal microscopy to track the movement of the particles. If you look at the top row, we saw that for 100 to 500 nanometers particles, sonication increased the movement of those nanoparticles by at least one order of magnitude. In the bottom row, we instead looked at the movement of individual nanoparticles. We saw that for the smaller nanoparticles, ultrasound enhanced the movement of almost all of them, shifting the entire curve to the right, whereas for the intermediate sizes, it was only a small subset of them. Our theory is that due to the heterogeneity of the tissue at baseline, sonication in turn produces heterogeneous effects and results in a range of pore sizes. Next, we wanted to confirm that in the increase that we're seeing in nanoparticle movement is a result of those very subtle ultrastructural changes that we saw in electron microscopy and not due to widespread damage in the tissue. And so we used tetrazoleum staining in H&E and saw that in the sonicated slices seen in the middle column, it was very similar to the control slices and sham-treated slices seen on the left, as opposed to what we saw in the positive control for mechanical tissue damage seen on the right-hand side. Most recently, we've started to translate our work in vivo using an MR-guided focused ultrasound system, and we used a raster method to first treat a volume of brain and then immediately infuse a compound into the region of interest, after which we could analyze its volume of distribution. This is still in the preliminary stages, but we're excited to continue our research into this area. Thank you.